next speaker will be Sherman from the University of Chicago.
Thank you. I would thank the organizers for inviting me, except I'm one of them, so we can dispense with that. This talk is going to be a little bit unusual for me because I normally like to talk about the newest things going on in the lab. But I made a decision that some older stuff we've been doing or have done, most of which is published, would be more interesting to this group. And that may not have been the right decision, but that's what I've decided to do. And the idea is to suggest some of our findings that might be relevant to those of you interested in modeling circuits, particularly in Cortex. So for most modeled circuits, particularly of Cortex, excitatory and inhibitory synaptic inputs are implied to be glutamatergic. Glutamate is the main excitatory transmitter in these circuits, and GABAergic, respectively. And each is generally modeled as if they're relatively homogeneous in function. However, glutamatergic inputs are not homogeneous. I'm going to emphasize that and why that could be important. And also, GABAergic inputs are not the only source of inhibition in Cortex and other circuits. So there may be other inhibitory sources that might be considered for modeling circuits. Many of the ideas I'll be talking about were formed in collaboration with Ray Guillory, who died last year, and this collaboration lasted quite a long time. So when I say we, I generally mean Ray and me. So for an outline, I'm going to start by talking about the basic circuitry of Thalamus, because this is where these ideas germinated. I'll then talk about the division of glutamatergic pathways into what we're calling drivers and modulators. I'll then talk about some examples of why this has proven, to us at least, to be a useful classification. Then I'll talk about some properties of the glutamatergic modulator pathways, their topography, the fact that they activate metabotropic receptors and what this implies. And then if I have time, I'm not sure how much I'll have, how the modulators control the gain of driver inputs, and also the tonic burst firing modes in Thalamus. So this is going to be the outline of what I talk about. Start with Thalamus. Thalamus is a paired structure of these red modules right in the center of your brain. They're strategic in the sense that virtually all information reaching cortex must pass through these structures, and every cortical area, as far as I know, gets an input from Thalamus. We can start off by considering the circuitry of the lateral geniculate nucleus, the relay of retinal input to cortex. This serves as a good model for Thalamus. In addition to the feed forward to middle layers of cortex, we're showing this as layer 4, there's a feedback from layer 6. We call this a feedback because this feedback innervates the same relay cells that innervate that column of cortex. And notice both of these pathways use glutamate as the neurotransmitter. They're glutamatergic. To complete the circuit, there's local GABAergic inputs from interneurons and cells of the reticular, thalamic reticular nucleus that provide inhibition of these relay cells. And finally, there's ascending inputs from various brainstem cell groups, cholinergic, noradrenergic, and so on, that provide classic modulatory control of the circuit. Now, this is a pretty good model for most of Thalamus. The main difference between the LGN and any other thalamic relay across species and across nuclei is the input to be relayed is different. So the non-retinal inputs are largely conserved. There are differences. I'm going to ignore those differences for the purposes of this talk and keep it simple. So if this were the somatosensory relay, this would be the medial lemniscus and so on. If you can identify this input to be relayed, you know a lot about the main function of a thalamic relay. I'm going to point out again that there are 2 glutamatergic inputs to these relay cells, and we've known for decades that they must have very different functions. If we want to ask the question, which of these inputs is carrying the information to be relayed by the LGN, we can gain insight into that question, the answer to that question, by noting the receptive field properties, because how a visual neuron responds to visual stimuli tells you a lot about the kind of information it can pass on to its postsynaptic targets. Now, the receptive fields of the retinal inputs have the classic center surround organization, and they are monocally driven, which means only stimulation of the eye from which the ganglion cell sends its axon, will activate this axon. Receptive fields of the layer 6 cells are much more complicated. They tend to be binocularly driven. They show a lot of specificity for things like orientation and direction, also spatial frequency, stereopsis, many features that are not seen in the retinal input. So the question is, which of these inputs is being relayed to cortex? Which is the information being relayed? And we know the answer to that by looking at the receptive field properties of the relay cell, and they pretty much match the retinal input. So we've known for decades that the retinal input provides the information to be relayed up to cortex. We call this the driver because, and when I use the term driver, I'm implying that that's the main information being brought into a cell or cell group. Partly we call it a driver because it powerfully drives the postsynaptic cell with very large EPSPs. So what's going on here? And this is a question that's still a bit open. But data from our lab and a number of labs have indicated that it's really has a modulatory function, much like the classic cholinergic, noradrenergic, and other modulatory functions. And as I'll show, it does affect the relay and gain of driver inputs. So the point here that I'm going to be building on is that even though these both use glutamate, these two inputs, their function is quite different. And we followed on to look at some of these differences. And that's what I'm going to emphasize here. I'm going to take a little detour to tell you about the techniques we and other labs have used to study these pathways. We start with slices from the mouse brain. These slices are about 0.5 millimeter thick. And we start with a slice first developed in Barry Connor's lab. If you cut the slice in exactly the right angle, and in this case we're looking at the somatosensory system, you can retain the axons in both directions between thalamus and cortex so that you can study these pathways. Many other axons are cut, and every angle of the slice you cut preserves certain pathways and not others. The nice thing about this is that the primary somatosensory cortex of the mouse and other rodents has what are called barrels in layer 4 that you can see in the living slice. Each barrel, in this case, is associated with a single vibrissa on the mystacial pad of the animal snout. And this is nice because you can not only tell where layer 4 is, but also where the primary somatosensory cortex is. Now, if the example here, if we want to study the thalamocortical synapse recording from a cell on layer 4, we can activate this pathway by electrically stimulating. But there's a problem if you just use this approach. And that is that these layer 6 cells that I talked about that have axons that innervate the thalamus have a branch that goes up to layer 4. So you can see that if you stimulate this pathway without specifying what you're activating, you don't know if you're activating the green pathway or the red pathway. This is a general problem with these kinds of experiments, and the way we have dealt with this is to use a technique of uncaging of glutamate, which has now become a fairly common technique. Basically, you flood the slice with glutamate, the neurotransmitter, but each glutamate molecule is in a chemical cage that makes it inert. So in this form, the caged glutamate has no effect on any of these neurons. You can then shine a precisely aimed beam of laser beam, and where that beam strikes these cage molecules, it breaks the cage and allows the glutamate to be released as if from a synaptic terminal. And the nice thing is that this glutamate now can only activate profiles that have glutamate receptors. So it will activate cell bodies and dendrites, but not axons, because these axons don't have the glutamate receptors. And by using this approach, we know that we're activating this pathway. And so this avoids the what we call the fiber of passage problem or the anadromic axon problem. If you want to complement the uncaging data, you can use very fine electrical stimulation now that you know exactly where the cells of origin are of a pathway. But virtually all the data I'll talk about can rely simply on the uncaging. So you can use this, for instance, to record a cell in cortex, activate the thalamic input with uncaging. You can record in thalamus and look at the corticothalamic pathways. You can look at local circuits in cortex. You can look at longer circuits between cortical areas using this approach. So it's a very flexible approach to study circuitry in these slices. Now, another slight detour is to talk about the postsynaptic receptors on cells, mostly on dendrites, that are going to be responsive to glutamate released by a presynaptic terminal. They come in two main flavors. Inotropic receptors, which include AMPA and NMDA, are protein molecules that live in the membrane of the cell. This is outside, this is inside. And when they come into contact with a neurotransmitter, glutamate in this case, there's a conformational change that opens up an ion channel. So the ion channel is part of the complex. And these ion channels are usually sodium. They could also be, in some cases, calcium. This allows a positive ion to flow into the cell, which would depolarize the inside of the cell and produce the EPSP, and it's a fairly fast process. Metabotropic receptors for glutamate come in actually 8 types, but we're going to talk about 2 types here, 1 and 2. And these are more complicated events. So you have a complex protein as the receptor that has as part of it a G protein. Now when the transmitter strikes the receptor, the G protein is carried away, it swims through the membrane and comes into contact with another protein complex. This is called the effector protein, and this leads to a whole cascade of second messenger effects. And these biochemical reactions can have many effects on the cell, including changing gene expression. It can have many, many things, but one thing that we've concentrated on, and many labs have, is the effect on ion channels, particularly potassium channels. And in this case, what this effect is of this second messenger properties is to close potassium channels. It prevents a positive ion from leaving the cell, and this leads to an EPSP. And notice how prolonged this is. It lasts for hundreds of milliseconds, or in some cases several seconds, and this depolarization lasts long after the initial event of the transmitter binding to the receptor. This is what you get with the first type of metabotropic receptor. The second type does the opposite. Instead of closing potassium channels, it opens them, allows potassium to leave the cell, and this leads to a long IPSP. So in this case, we can see that glutamate attached to the correct receptor can actually inhibit cells, and for a very long time. Another important difference has to do with the location of these receptors. The inotropic receptors tend to live right at the synapse, whereas the metabotropic receptors seem to be further away, usually seen as forming an annulus around the synapse. And one thing this means, or this is the explanation for, that inotropic receptors respond even to single action potentials. So the transmitter is released and it'll affect these receptors. But to activate, so any frequency of firing here can get you an AMPA response. But in order to activate metabotropic receptors, typically you have to have firing in the axon of greater than about 10 hertz. This can vary, of course, in order to release enough transmitter to reach the more distant receptors. What this means is that this axon, if it's not firing very much, will only activate inotropic receptors. But as it becomes more interested in life, it'll start bringing in a metabotropic component. The higher it fires, the more the metabotropic component plays a role in the responses. So to show how this all works, this is a slice preparation, it turns out, from the cat. We're recording from a relay cell in the cat lateral geniculate nucleus. And what I'm going to show you here has been demonstrated in rodents and primates as well. If you activate the modulator input from layer 6, you see this pattern of activation. The EPSPs, as you note, get larger as the train of stimulation goes on. This is called a facilitating pattern, and it's correlated with a very low probability of release of the at the terminal of transmitter. Each action potential has a low probability of releasing transmitter. You also note that as you increase the power or the electricity here, you increase the size of the EPSPs. And this suggests that as the current spreads, you're bringing in more and more axons, and this is consistent with a lot of convergence in this input. As long as the stimulation is at low frequencies, about 10 hertz or so, you can knock it out completely by using AMPA and NMDA receptors. And with these receptors still in place, you can demonstrate the metabotropic component of this pathway by a burst of about 10 shocks at 50 to 100 hertz. So it's fast enough to release enough transmitter, and you get this very long, slow EPSP. Notice the time course here. And if we now add to this a specific metabotropic glutamate 1 blocker, you knock that out. So this is the metabotropic component from this pathway. We can then, from the same cell, look at the driver input, and we see everything is different. Much larger EPSPs, activated in an all or none manner, as if there's very little convergence in this pathway, and they depress. They get smaller instead of larger as you go through a train. And this is correlated to a very high probability of release at this synapse, which is, these are properties you'd want in a driver, and there's no metabotropic component, only ionotropic components. Okay, so to summarize these differences, I'll use a thalamic relay cell to start. The driver inputs, there's some anatomical correlates here as well, innervate proximal dendrites in complex synaptic zones known as glomeruli. They activate only ionotropic receptors, and I've already talked about the activation profile. Modulators have more convergence, and they innervate distal dendrites. They activate metabotropic as well as ionotropic receptors. Again, the activation profile I've gone over. But there's also other anatomical differences. The drivers tend to arrive over very thick axons with small and large terminals and very dense arbors, whereas the modulators have very thin axons, strictly small terminals, and very sparse arbors. So all these differences that we see in thalamus apply pretty much in cortex as well, although the data in cortex aren't quite as good, with the exception of the glomerulus. The glomerulus is a thalamic feature. It's not found in cortex. But numbers are also important here, because if you consider all the synapses on the relay cell, the driver inputs represent less than 5%. These glutamatergic inputs represent an order of magnitude more synapses, which is surprising if we're calling the driver input the important one for information transfer. The rest of the inputs come from GABAergic and classic modulatory inputs. But it's interesting that these numbers are also seen at the first stage of processing in cortex. So thalamocortical inputs to layer 4 cells, which are the drivers, represent about 5 to 6% of the synapses, and the modulator inputs that have been identified on these layer 4 cells represent about 40 to 50%. So all these features seem to apply to cortex as well, including the majority of glutamatergic inputs being modulators, although the numbers in cortex are much more limited. The examples are more limited. But this is a general pattern of some of the differences. We've identified these two kinds of inputs in mouse brain slices onto a number of examples onto thalamic cells, activating different kinds of inputs to them. I'm not going to go through this in detail. And onto cortical cells. I'm just making the point that what I'm going to show you in the next few slides is based on examples from a number of different thalamic and cortical cells and activating different kinds of glutamatergic inputs to them. So if we look at a table of differences, there are 10 or 12 of these parameters that we've identified that we believe largely to be independent of one another. We wanted to show how these distribute in scatter plots, and we picked 3 of them to quantify. I think any 3 would give us the same picture. The 3 we've chosen are these 3 here, the details of which aren't important, but when we plot these, we get scatter plots that look like this. Each point here represents a single cell from which we've identified glutamatergic input as driver or modulator onto the cell, and the color code tells you whether the cell is in thalamus or in cortex. And there are a couple of points to make from this. The difference between drivers and modulators is quite robust, and I hope you're convinced by this alone that the classification does tell us that there are different kinds of glutamatergic input. Second point is we only see 2. Frankly, when we got into the more complicated circuitry of cortex, we expected to find more classes. So far we haven't. Maybe as people explore other areas of cortex or other parts of the brain, there'll be other kinds of glutamatergic synapse identified. The third point is that the modulator properties onto thalamic and cortical cells are pretty much similar, as are the driver properties, as if we have 2 basic kinds of glutamatergic synapse that operate in thalamus and cortex. The hypothesis is that drivers are carrying the main information, and modulators affect how the driver inputs are processed. This hypothesis is pretty strongly supported, we believe, in thalamic circuits. In cortex, I think the jury's still out. We need more evidence for cortical circuitry. It is more complicated, but the evidence that does exist is consistent with this hypothesis, but it's far from proving it. I'm going to provide a couple of examples of how using this classification changes the common or textbook view of the functional organization of circuits. I'm going to start by throwing out something that Ray, Guillory, and I have worked on to show how cortical areas are connected, because this will be important for a couple of the examples I'll be showing. If this is the visual system, this is primary cortex, and we can move up the hierarchy. We've known for a long time that these areas are connected directly with each other. But what Ray and I have emphasized is that there's a parallel pathway to connect these areas through thalamus, in this case pulvinar. And it does appear to be the case so far that when cortical areas have a direct connection, there's what we call a transthalamic one that also goes through the thalamus. So there's something interesting here. All cortical relays get a feedback from layer 6 that is glutamatergic and modulatory, but these pulvinar cells, in addition, get a cortical input from layer 5 that is feed forward and driver, because the synapse here has exactly the same properties as the retinal input to the lateral geniculate nucleus. This duality we call the LGN an exemplar of a first-order relay, because it's a first relay of a particular kind of information, a cortex, and pulvinar a higher-order relay because it's relaying information already in cortex between cortical areas. And if you're asking about bandwidth, the pulvinar is about 30 to 50 times larger in volume than the LGN, so there's a lot of bandwidth for this to go up the hierarchy. This duality applies not just to the visual system, but the somatosensory system has VP as a first-order relay, the posterior medial group is higher order, the ventral part of the medial geniculate nucleus is first order for auditory processing, the dorsal division higher order, and so on. And the medial dorsal nucleus, which is the second largest relay in thalamus, we see as mostly, if not completely, higher order, connecting areas of prefrontal cortex. So we believe that most of thalamus, by volume, provides this kind of higher order function. Okay, with that in mind, I want to consider textbook views of how sensory information reaches primary and higher areas of cortex. So, and the idea is that there are parallel routes of information from the periphery. So for the visual system, the idea is that one pathway goes through the LGN to primary cortex, but there's a second pathway in parallel that goes from the superior colliculus and pulvinar to cortex. And we see the same in the auditory system. Two parts of the inferior colliculus, the core and shell region, have these parallel inputs through what we're calling first and higher order relays. And for the somatosensory system, we get parallel processing from the principal nucleus of the fifth nerve and the spinal nucleus of the fifth nerve. This would seem to be somewhat at odds with what I'm telling you, that these higher order relays are not used for a parallel subcortical input to go to cortex, but are rather used in these transthalamic cortical pathways. So to address this, we start with the auditory system. We used a slice preparation that preserved the connectivity between the inferior colliculus and the medial geniculate. We could record from a cell there, do our uncaging in a grid over the colliculus, find the hot spot where the cells that provide excitatory input, these are inward currents. Here's a cell we recorded from. Then we could test these synapses for whether they're driver or modulator. For the lemniscal pathway, which is the core of the colliculus to the ventral part of the medial geniculate, every single cell showed a driver pattern, depressing synapse and no sign of a metabotropic receptor. For the non-lemniscal pathway, from the shell of the colliculus to the dorsal division, we found the opposite. Every cell here, and it's quite a dramatic difference, showed a facilitating synapse and a type 1 metabotropic receptor component. And so we suggest that this textbook view of what's going on here is not correct. Instead, what's going on is that this so-called parallel pathway is really providing modulatory actions on the transthalamic pathway. And this, again, if you follow the logic here, using the driver modulator distinction does change the way you look at the functional organization of the circuit. We can take this to other examples here. So for the auditory system, we suggest this is what's going on, and we see a very similar thing from a paper we just published. And the somatosensory system is a little bit more complicated, but it's pretty much the same. We don't know yet for the visual system. This is on our list to test. One final example has to do with the basal ganglia. I believe there's some interest in basal ganglia here. When I look at textbook views, they show the basal ganglia as part of an information loop, that you follow the information from cortex through the basal ganglia to thalamus and back up to cortex. And we think there's a problem with this that has to do with the nature of the basal ganglia input to thalamus. So if we look at the motor thalamus, which is mostly VAVL, a complex of ventral anterior and ventral lateral nuclei, we see two inputs that are relevant, one from the deep nuclei of the cerebellum and the other from the basal ganglia. The problem is the basal ganglia input is strictly GABAergic. And we've argued that every thalamic nucleus has a driver input that represents the information to be relayed, and that driver input is glutamatergic, which suggests that this is not an information route, if that's correct. And we can parse this because this VAVL complex is really a mosaic. And what's interesting is that data in the rat and in the monkey has shown that the cerebellar input and the basal ganglia input avoid each other. They each innervate different territories of this mosaic. Furthermore, there is a layer 5 input to this structure, and it seems to target strictly the region of VAVL that is innervated by the basal ganglia and avoids the cerebellar input. So it looks to us, and what we're suggesting, is what the basal ganglia is doing, just like the previous examples of the sensory pathways, is it's modulating, affecting the gait because it's GABAergic of the transthalamic pathway. And again, that's a somewhat different view of how the basal ganglia relates to cortex, that it's not providing basic information to be relayed, but is doing something else here. So an example could be seen as follows. Imagine you have a cortical area that has direct connections and transthalamic ones to a number of other motor cortical areas. If the basal ganglia input is active here and here, that leaves only these two pathways, these two connections with other cortical areas as having both pathways intact. And the pattern can change depending on which nuclei are getting active inputs from the basal ganglia at that time. This can be seen as a very dynamic process. Now, what this actually means, I can only speculate, but one possibility is that thalamic input to a cortical area is necessary, not necessarily sufficient, but necessary for the columns in that cortical area to function properly. And if I were talking about new data from our lab, I'd provide some data to support that. And that means that these areas that aren't getting its thalamic input anymore are not functioning properly. And that means that only these areas are connected functionally with the primary area here. It may also be that there's some coincidence detection going on that's very important for the functioning of these areas. But in any case, this is one idea of what the basal ganglia is actually doing, which I think comes from this primarily from the driver modulator distinction and is still not seen in textbook versions of this. Okay, now I'm going to talk about some properties of these glutamatergic modulators and try to finish up quickly. You might ask, since the thalamus gets all these classic modulators, why is there a glutamatergic modulator added on? And we suggest that it has to do with topography. The cholinergic, noradrenergic, and so on modulatory inputs are very diffusely organized. For things like focal attention, adaptation, anything where you just want one or two cortical areas to be affected, you need topographic modulation. Because if you depend on cholinergic, for instance, modulation, you modulate the entire cortex the same way when you only want certain small areas to be affected. So you need to have topographic modulation, and the glutamatergic inputs are the only ones we know of that can do that. So if you look at all the classic ones, this is from a textbook, you see that they're very diffusely organized with very little topography, but only the glutamatergic ones provide the modulation that is highly topographic. And so we think it's necessary for for many of the functions I've suggested, and you can imagine many others. I have two more points I'd really like to have time to go over, but I'll start with what happens when these metabotropic glutamate receptors are activated. So here's an example, again, of a driver input. It's the thalamocortical input to layer 4, a depressing synapse, no metabotropic component. Here's a modulator, layer 6 cortical input to a thalamic relay cell. I've already gone over this, facilitating synapse and a large depolarizing metabotropic response that can be blocked by a type 1 blocker. But we have another example here, and this is the layer 6 to layer 4 modulation. Here, you get the strong IPSP that lasts for a long time. So you can get both excitation and inhibition depending on the receptor, and this is blocked by a type 2 blocker. So to show this, the importance of the staining, this is immunocytical staining for this type 2 receptor, and you can see it's in all these three sensory areas. It's very densely found, particularly in layers 4 and 6, but in other layers as well. So from this and from our recordings, I would argue that the type 2 metabotropic response that provides inhibition is pretty widely distributed in cortex and certainly shouldn't be ignored. So again, if we focus on this branch of the layer 6 cell that innervates layer 4, we can study that by uncaging, record from a layer 4 cell, uncage, and you see that there's a region here that provides some inward currents. So this is the connection. This is just a false color view of the hotspot here that provides input to layer 4. If we do uncaging at a high rate, you see the inotropic response here, but we can ignore that. This is the metabotropic component. And we know that because if we put the mGLUR1 block in, we reduce that. If we do a subtraction, you can see what the metabotropic component is, and it's fairly substantial. This is another example where we're recording from a layer 4 cell. The hotspot is here under the triangle, but here we get a different response. Here's our inhibitory metabotropic component. We see it when we wash it away, and this is due to a type 2 metabotropic receptor. We also see this in cortico-cortical connections. Here we see a driver input from V1 to V2, layer 4 to layer 4. Here we see a modulator from layer 2-3 to layer 2-3 that has a type 1 metabotropic response. But here's one from layer 6 to layer 5A that is type 2. So we see these inhibitory responses in a variety of different cortico-cortical circuits. The final main point I want to make, and I think I can quit after this, is using these metabotropic receptors and the modulatory pathways to change the gain of driver inputs. So in this experiment, we're recording from a cell in layer 2-3, and the uncaging shows hotspot in layer 4 just below it, as well as a smaller hotspot onto each side, and it's shown here. You can see the lateral hotspots and the larger one just below it. So you can record from the cell and activate these two inputs and see how they interact, because as we'll see, the layer 4 input is a driver, a depressing synapse, no metabotropic component, whereas the layer 2 lateral input is a modulator, facilitating synapse and a large type 2 metabotropic component. What happens when we activate the metabotropic component to see what effect it has on the driver input from layer 4 to layer 2-3? And that's what we're going to look at here. Again, we're recording here. When we just stimulate layer 4, we get this pattern, the depressing pattern. But when we do the stimulation after activating the modulator input, remember the action can last for hundreds of milliseconds to several seconds after you stop the stimulation. The EPSCs that are evoked seem smaller than here. But if we do this with a metabotropic blocker, the same experiment, we don't see any effect on these EPSPs. And it's easier to see if we take them apart. This is the effect, a large reduction of EPSPs when we activate this modulator pathway. But when we activate it with the type 2 metabotropic receptor block, we don't have any effect. So to summarize, we've seen this in the layer 4 to layer 2-3 in both visual and auditory cortex. There's a reduction in this synapse when you activate the pathway. from V1 to V2, the feedback from higher auditory or visual areas to primary in layer 4. The input from LGN by this pathway is knocked down. The layer 6 input to LGN, when this modulator pathway is seen, you can knock down the retinal input. But what's interesting, if we go back to this and we look at the modulatory input alone from layer 6 onto these cells, there is no effect on activating the modulator pathway. Now, we'd like many more examples to see if there's a real pattern here. What we can say is in many cases, driver inputs have their synaptic strength affected by the modulator inputs to the same cell. We haven't found yet an example where that's true for modulator to modulator inputs. But again, this is early stages and we need a lot more examples. This last point I'm just going to go over very quickly and just make a point. I'm not going to go through the actual data. This is for those of you who really care about thalamic relay cells and the fact that they can be activated in either burst or tonic manner. And these response modes are very important to the nature of the relay to cortex. And all thalamic relay cells show this. These response modes are controlled by voltage-gated conductances, and we and others have shown that those voltage-gated conductances are controlled very effectively by the layer 6 modulatory feedback. And if you, by activating the layer 6 feedback, you can very clearly and precisely control which response modes these relay cells will be firing in. I'm happy to talk about that in detail with anybody offline. I don't have time to go over it here. Okay, so to summarize, the driver modulator classification leads to a very different way of looking at glutamatergic circuit properties and information transfer. Modulators appear dominant in thalamic and cortical circuitry. So if you just do a connectomics approach and just count up all the synapses you have and assume that the pathway with the largest number of synapses is the important one, I hope you realize that that could be very misleading. Glutamatergic modulators provide topographic modulation, which we think is a very important feature of them, and the main thing that distinguishes them from the classic modulators. mGluRs are ubiquitous in cortical and thalamic circuitry. If you need a prolonged process that takes up to several seconds, think of metabotropic glutamate receptors. Activation of mGluRs can produce excitation or inhibition, depending on the receptors involved, and the inhibition can be quite profound and long-lasting. Activation of mGluRs can also affect the gain of driver inputs, at least in some cases. And as I've said very quickly, it also controls the burst firing mode in thalamus. And I'll leave this with a question. How important is it to incorporate these concepts of modulators and drivers and models of cortical circuitry? I don't know the answer to that. It may be that this isn't terribly important yet with how well we are able to capture the functional properties of cortical circuitry and circuitry in other parts of the brain. But the purpose of this talk was to alert people to the importance of this in getting a detailed understanding of how these circuits operate. Thank you. Yeah.
I'm wondering if there's the same classification can be applied, which I mean it's receptor-based, so it can be applied to lower level circuitry, in particular the output circuitry from the neural system, in particular the actuator of chemical glands and muscles. So is there any work done in this direction, any classification?
Great question. How useful is this outside of thalamus and cortex? I can only say it's a great question. I think we need to know the answer to that. And I think that, let me put this in some context. When you go in to study a new structure like the retina or cortex, one of the first things you will need to do is classify the different elements there. So to understand retina, for instance, you need first to know that there are rods and cones and ganglion cells, which is maybe equivalent to the level I'm talking about to different transmitter systems. But now we know we have many different classes of ganglion cells and amacrine cells in retina. And that's analogous to many different classes within transmitter systems. talking about this for glutamate. And I think the classification of circuits based on transmitter systems hasn't really been done very systematically. The cholinergic pathways are all the same, and so on. I think that question has to be, there has to be a proper classification to see how many types of cholinergic input there are. And this could apply, as you suggest, throughout the nervous system, peripheral as well as central.
So your 3D plot seems to suggest there's some correlation between, on one hand, postsynaptic metabotropic receptor expression. On the other hand, whether synapse is facilitating or depressing.
That's right.
How could that be? How are those two things?
The facilitation and suppression applies just to the inotropic responses, not to the metabotropic. So I don't see any problem with that. They're independent.
So the metabotropic response is postsynaptic, right?
Yeah. But the-
whereas
but-
when
but-
the synapse show facilitation and depression.
The facilitation and depression, the facilitation and depression is defined strictly on the basis of the responses of the inotropic receptors. So the fact that a cell can be either, it turns out that the cells that show suppression, facilitation are the ones that have metabotropic receptors. That's an empirical observation. I don't particularly see a problem with that.
Well, let's say this particular neuron that has metabotropic receptors, right? But it gets input from different pathways.
Mm-hmm.
Imagine that some pathways show facilitation,
Right.
some others show depression.
That's right. So we're looking at a specific pathway that shows only one or the other. Now, I don't know if this was your question, but why don't we see more evidence of activating pathways that show both? And so you can imagine activating a pathway that has both drivers and modulators as an example. Maybe I should have mentioned this, but one surprise to us is that we just don't see that very often. When we do, it's very obvious. And to make sure that we have artificially, or not artificially, we activated known pathways to a cell that are both, one pathway is driver and the other pathway is modulator, just to see what that would look like. And it's very easy to see that. So when we activate these pathways the way we've done, we see almost no examples, there have been a few, but almost no examples of mixture between drivers and modulators. That was a surprise to us. I'm not sure what to make of that, but they do seem to be separately organized in terms of where the inputs are coming from.
I have a question about GABAergic neurons. So, do you think there could also be driver and modulators there? And also in the thalamus, do you have lots of interneurons, and do they have different targets like soma and dendrites and something?
So GABAergic inputs, we are asserting, this can be challenged, this is pure speculation, cannot be drivers. The drivers have to be, by our claim, glutamatergic, a subset of these glutamatergic inputs. One reason is that if you try to model the amount of information you can get across an inhibitory synapse, it's much lower than excitatory synapses. It has to do with the fact that, well, we can talk about that offline. This has been modeled. So GABAergic synapses, it turns out, are very inefficient in getting information across a synapse unless the postsynaptic cell is firing at very high rates, more than about 200 hertz in our modeling.
But there is...
Now, your other question is about the termination on postsynaptic cells.
because,
we-
you know, the PV versus some cells in cortex.
Cortex is very complicated.
Well, I'm oversimplifying here. Some target dendrites, and some could control modulator inputs, maybe.
Some go for the initial segment of the axon. I mean, they're all, so the different GABAergic cells in cortex have very different innervation patterns on the postsynaptic cells. In thalamus, we see that too. On the relay cells, there are two main GABAergic inputs. The local interneurons innervate proximal dendrites. The TRN cells innervate distal dendrites. And there's no overlap in their territories. We can speculate about what that might mean, but in cortex, as you know, it's even more complicated than that. But very specific targets for the different classes of GABAergic cell, and we see that in thalamus also. Yeah.
Do you know the facilitation?
Don't hear you up here.
Facilitation and modulatory pathway, is it presynaptic or post?
Is it
Presynaptic or postsynaptic?
the facilitation or depression is thought to be a property of the probability of release of transmitter. And that in turn seems related very closely to the size of the calcium conductance that is generated in the presynaptic terminal through the invading action potential, whether it's active or not. And this has been modeled quite nicely, and things that allow you to change the calcium conductance by changing how much calcium there is in the extracellular fluid all support that idea. So to step back, if the calcium conductance in the terminal is relatively small, you get less probability of transmitter release, and that will lead to facilitation. If it's big, you get the opposite. So it seems to be best explained strictly on presynaptic terms about how much transmitter is being released. Yep.
Can we assume that the modulators are basically transferring much less information? They have less bandwidth than the drivers?
You're asking about modulators versus drivers?
Yeah, in terms of bandwidth, is the information flow through the drivers of much larger bandwidth than the information flow through the modulator?
I'm not sure what you mean by bandwidth and drivers.
Think about like how many bits per second you're transferring essentially through the drivers versus the modulator.
Right. So if you apply information theory for that, I think what you'd need to do would be able to record pre- and postsynaptically simultaneously. That's been done for driver inputs, retina to geniculate, and in some cases thalamus to layer 4 and cortex. I don't know of anybody that's successfully done this with modulators, and here's the problem. Let's take the LGN as an example. The LGN gets input from 1 to 3 retinal ganglion cells, so it's possible to do the paired recording and see a big effect. We estimate that the layer 6 input to these same cells, the modulators, have at least 100, maybe several hundred, converging axons, each of which produces only about 10 synapses. If you did a paired recording there, you'd have such a weak response that it would be very difficult to see. People have tried, and they report at best some very weak responses that are so hard that people have pretty much given up trying to do that experiment. So I don't know if anybody's applied information theory for the driver synapses, but the modulators, no, I don't know of anybody that's done that. I just think it hasn't been done. It's technically just too difficult.
Yeah, so can you comment about TRN and like, so layer 6 synapse to TRN, that's also a modulator? And,
Right, so TRN gets inputs from axons that go through the TRN. They include the layer 6 cells that come down to innervate the thalamus and the thalamic relay cells that go up to cortex. Both provide branches. If you're asking which of those is driver, that's not been clearly, that's a good question that should be addressed. It hasn't been clearly addressed yet. The best guess, based on weak and direct evidence, is that the relay cell is providing the driver input and the cortical, the modulator, and that's based strictly on the size of the synapses that are formed. And that's only one of these parameters, and I'm not sure how reliable that is in this case. So that's really still an open question, I think. Yeah.
In your picture, or let's say if I can call it that, a paradigm, you basically have, in your paradigm, you have a circuit with information flowing in it, which has modulators and drivers, which you can then block and therefore identify drivers versus modulators. What and basically the whole discussion goes in that, and it's with information flow. I mean the discussion runs in terms of information flow. I'm wondering, just to kind of make the discussion bigger, if one said, okay, I'm not allowed to talk about, well, basically the circuit lines that you're drawing are huge wires from area to area. But of course, if we look at Professor Moss's picture or something, we would say instead those interactions are driving the search in a very complicated space statistically in a different way. Or one could also say, you're modifying parameters that make an attractor network go somewhere else. And so the only thing I'm throwing out is when, for wider discussion, is whether that circuit model, assuming information flow, is really going to work according to the theorists. So in other words, if you have feedback, can you use information flow as your basic concept? If you're showing your emphasis is feedback, that there's a strong feedback.
Yeah, I'm not sure I follow the reasoning there. If you believe in a hierarchy of cortical areas, isn't the information going, let's say, from V1 to V2 information?
If you have a highly connected feedback loop, I don't know if you can call that where you massively expand the space of the neurons working. I don't know if you can use the concept of information.
Okay, I'm out of my depth here.
I
So
threw that out for theorists.
I don't have any insight there.